Thank you so much, Ben. Close, it's Inosine, but that's okay. It happens to the best of us. Also, I can't see you guys, but thank you so much for coming to my talk tonight. I am a rising fifth year at Emory, as Ben said. I'm in the Department of Chemistry under the direction of Dr. Jen Heemstra. And I would love to talk about my most recent publication, which is Systematically Modulating Aptamer Affinity and Specificity by Guanosine-to-Inosine Substitution. So that was a really big title, right? And I know when I first got to grad school, I had never heard of an aptamer and I had never heard of inosine. So I would love to talk about first what aptamers are. Aptamers are simply single-stranded nucleic acids. So this can be DNA or RNA or some specialized nucleic acid that can bind a specific target. In our case, we study small molecule aptamers, but this target can be anything from a small molecule to a protein to a receptor. And so basically, you have this single-stranded nucleic acid, and in the presence of a target, it adopts this secondary structure and binds with high specificity and affinity typically. But one thing that's kind of I guess difficult when working with aptamers is actually generating them. So we don't really have aptamers just present in nature, especially not DNA aptamers. And we typically want to go towards DNA versus RNA because it's much cheaper and is a lot more stable in nature. So whatever your function is, or whatever your application is for your aptamer, usually biological, you want something that's going to be rather sturdy, right? And so a lot of us know RNA is a lot less stable than DNA. And so how do we generate these aptamers? We use CELEX, which is systematic evolution of ligands by exponential enrichment. And so we start off with a single-stranded DNA or RNA library. And this library consists of this region that's usually in the middle that has usually N20 or N40 random sequences. And so as you can imagine, you get a very diverse library of about 10 to the 14 sequences. And some of them will be able to bind whatever your target is, but most of them won't. And so how do we select for this desired function? We take our target, in our case, as you guys know, is going to be cocaine, but we take our target and we introduce it to this library. And we get some sequences that are active and we get some that are inactive. We isolate our active sequences, getting rid of the inactive, and we carry it for another round of CELEX. Now, how do we generate really great aptamers? Well, we increase our stringency upon every round. So let's say we start off with 1 millimolar of cocaine whenever we introduce it to our target. And let's say we get 500 sequences that bind. The next round, we carry forward those 500 sequences. And this next round, maybe we're going to do 750 micromolar of cocaine. And of that 500, let's say 20 bind. And as you can imagine, right, each round, we just keep on increasing that stringency until we get the best sequences. And then we go forward and we identify and characterize our candidates. What I've learned in grad school is that CELEX never really goes how we want it to. One big issue with CELEX is you're not only selecting for what you want, you're also selecting for what you don't want. So there's that other added component of just being sure that you're selecting, an aptamer specifically for cocaine. And so, as you can imagine, generating aptamers isn't super straightforward. So what people try to do now is once they have an aptamer in literature, what if this aptamer doesn't bind with the affinity that they want? What are ways that we can modulate our aptamers to bind with a better or a worse affinity, right? Because once it's generated, it's generated. And you get your aptamer and you get your nice little binding curve. You get a specific KD here at the midway point. Right now in literature, some of the most common ways of generating aptamers from an already known aptamer is usually by truncating. So if you see here in this stem, we chop it off here, or by having crucial base changes. So of course, we are doing G to I base change. But when I say crucial, I mean a large area of base mutations. Or instead of G to I, going like G to A or A to T, something that's a little more drastic. But the big issue here is that instead of getting a range of binding affinities or a better or worse affinity, you may get no affinity at all. You may completely perturb your binding. And so just to preface, this is something I was telling Ben before we started. What's really fun about this project is that it wasn't an original project that I came in on. We actually were trying to generate aptamers for cocaine with a range of binding affinities. And the way we thought about doing that was using Inosine in place of guanosine. And when we recognized that we could get a really wide range that we got, we went forward with this from my main project, and we explored a little bit further and just, got a like kind of a small paper out of it. So that's been fun with that as well. And so again, our target is cocaine. And so we went to literature, and the MNS 4.1 anti-cocaine aptamer is the first cocaine aptamer generated. And from in literature, what some people have done is they've gone in and made 38GC and 38GT. So basically all they did, we're going to focus on what's in yellow for what's in literature. All they did for 38GC was in STEM 1, they changed the ACAG to CCCT, and this GT base pair here to GC. And in 38GT, they only changed what's here in STEM 1. The issue that we ran into here is that the difference in KD or binding affinity was only about 2 or 3 different in KD. And so we were looking for a wider range. And so we thought, well, how can we do this without, again, severely perturbing that binding? And of course, as you guys know, we substituted key guanosine residues for inosine, because inosine simply has one less amine than guanosine. And we just didn't really think that this would severely perturb that secondary structure. So now we're going to look at what's in pink. Pink is going to be inosine here. Fun fact, pink is my favorite color, but that's not why I chose it here. I actually chose it for colorblind friendliness. But yes, pink is my favorite color. And what we did with our G to I's is in all of our aptamers, so we kept the parent aptamers, MNS 4.1, 38GC, and 38GT, and we added those in with our pool, but we generated more. In STEM 1 of all of them, we changed Gs 1 through 3 in STEM 1 to I. We chose these Gs because we read in literature that STEM 1 is crucial for cocaine binding. And a lot of times in literature for the cocaine aptamer specifically, they truncated this stem and completely lost binding of cocaine in general. And so going forward, any change to STEM 1 is going to just have inosine after the name. So I3 would be MNS 4.1 inosine, 38GC inosine, 38GT inosine. And I will keep bringing this picture back up throughout the talk as well. We also went into G25. The reason we went to G25 is because cocaine binds around here, and we did not want to get too close to the binding pocket. So in 38GC and 38GT, we changed G25 to I, and that would be I25. Those got named 38IC and 38IT. So G25 would just change the name from G to I. Using microscale thermophoresis, we were able to test the binding affinity. The reason I want to talk about this technique is because it's a rather new technique. I would say maybe 10 to 15 years old. A lot of people, when they test binding affinities of any two molecules, they use typically ITC or SPR, but we went with MST because we can use smaller amounts of it, or small amounts of our targets, and it's a lot quicker. So cocaine loves to hydrolyze in solution on its own, and we didn't want to lose our concentration of cocaine by doing ITC because those runs are typically a little bit longer, 2 to 3 hours at times, and we really didn't want our cocaine to hydrolyze while we were trying to run our measurement. So we just went to MST. How MST works is studying thermophoresis. Molecules that are heated up want to leave the heat and move towards what's cool. So let's say the laser here, they use an IR laser to heat it up, is hitting this end of the capillary. All of the molecules that are in the presence of the laser are going to move away from it. So we are studying the movement of these particles due to heat. Because our aptamers are labeled with Psi5, we are able to look at this movement by looking at the loss in fluorescence of where it's being measured. Because again, those molecules are moving away from the heat. When the laser is not on, or our initial state, we have 100% fluorescence. Over time, again, once you introduce fluorescence for a set amount of time, we start to lose our fluorescence intensity because, again, those particles are moving away. So our Psi5 labeled aptamer is moving away from the laser. So we get to see that loss in fluorescence intensity. But how can we tell what's the difference between bound and unbound, right? Well, the smaller the complex is, so aptamer only, for example, the faster it's going to travel in that set amount of time. And the more cocaine you add, the more binding you get, and therefore the heavier it is. and you get slower drop in fluorescence. And that's how we're able to test the binding affinity using MST. And so, of course, we start with our parent target, right? Cocaine. And again, the objective I want to remind you guys is to generate aptamers with a range of binding affinities. So we're not necessarily looking for a better aptamer or a worse aptamer. We just want to see if we can use a quicker method, a more robust method, to give us a range of affinities instead of going all the way back through CLEX or trying to truncate with trial and error, right? Going forward, we're going to look at our Mst binding curves. For those who are not familiar with binding curves, we are looking for this midway point, which is our dissociation constant, or our KD. In other words, this tells us when half of our aptamers are bound to whatever target. In this case, we're looking at cocaine. And what we see is that we actually do successfully generate aptamers that bind with a range of affinities. So again, MNS 4.1 was the parent aptamer. This is the original aptamer. It has the lowest affinity for cocaine versus all of our enzyme binding aptamers. And we do get a range of about 80 micromolar to about 230 nanomolar. So we were really excited to see such a drastic change because, again, in literature, we were only seeing a change of like 2 or 3. But this is where it gets fun, right? When we saw that the change was so drastic, we actually went forward to look at other off-target effects. In literature, it's shown that the cocaine binding aptamer not only binds cocaine, it also does not bind EME or BE. EME or BE are basically just degradation products of cocaine. EME is generated when cocaine is hydrolyzed by the bacterial cocaine esterase, and BE is generated if we ever do cocaine. I hope we don't do that, but in our bodies, if we ever do cocaine, our carboxyl esterase will degrade it into BE. And so because we knew that the parent aptamer didn't bind EME or BE, we were really curious to see if changing G to I would actually restore this affinity. And just showing 2 of our aptamers here, our 38GC inosine, none of our aptamers bound EME or BE at all. So we were really excited to see that, just because it was something that was really cool to pick up from it. But we didn't stop here. We also wanted to determine whether or not inosine and guanosine substitutions would affect off-target recognition of known binders. So 2 other analogs of cocaine are norcocaine and cocaethylene. Both of these are known to be bound by the parent aptamer. And so we decided to see if we could also tune this recognition. So starting with cocaethylene, we chose cocaethylene not only because it's a known binder, but also because if someone were to do cocaine and also drink alcohol, they will also have a presence of cocaethylene in their drug test. So it was something that was also very much so biologically relevant. And even here again, right, we're able to see a range of binding affinities, again, not only for cocaine, but now it's off-target or its analog, cocaethylene. And then we did norcocaine because it is the only pharmacologically relevant analog of cocaine. And because, again, right, the parent aptamer in literature is shown to bind norcocaine. So we wanted to add another analog to see if we could also tune that recognition. And we are able to tune it. And we're also able to, again, lose that affinity for this off-target with one of our other inosine-containing aptamers. And so up until this point, not only have we shown that we can get a range of binding affinities for cocaine, we show that we can get a range of binding affinities for its off-targets too, right? Which was also really cool because if you ever wanted to look at someone's drug test, and let's say cocaine over time has hydrolyzed to where you're out of the limit of detection of your biosensor, if this method would be shown to work and they had done alcohol as well, the cocaethylene may have been higher, or maybe it wasn't higher. You have an aptamer that may have a high enough affinity, right, to where it can sense the cocaethylene, even if the cocaine is no longer present at a detectable rate. And so we are not a structural biology lab. We don't do crystal structures of our macromolecules and study them. But we still wanted to investigate what could possibly be the reason for this range of binding affinities. And so the question we asked was, how does G to I affect thermal stability of our aptamers? And, just old-fashioned TM studies, right? So bringing back up our picture and a lovely table here that I promise I will break down for everyone, we don't actually see a trend in our TM. studies. But something I do want to note, one small trend, we don't see a large trend, but one thing we do see is that if we have our parent aptamer, right? So MNS 4.1, 38 GT, again, no position G to I, and 38 GC. Those that we changed the G1 through 1, 2, and 3 to I, so 38 GT inosine and 38 GC inosine, these were the only two where we saw a drastic decrease in our TM from G to I, or from parent to a more drastic change in base modifications. We also see the same here, right, with 38 GC and 38 GC inosine. While I'm not sure why that may be, definitely more studies would have to be done to determine this. It is consistent with literature because, again, something that's seen in literature is truncating this stem altogether not only decreases the stability, but it also actually completely diminishes the binding. So we have a method where while we are decreasing the stability of our aptamers, we are not completely getting rid of the binding. So that's why G to I was really the way for us to go, because again, if we truncate, we won't bind at all, versus changing the G to I, we were able to get a range of binding affinities. Even if we were decreasing the stability, we were still able to produce aptamers that bind. But of course, this paper is in analytical chemistry, and it would not be an anochem paper if we did not look at the biosensing ability of these inosine-containing aptamers. And so then our next and our final question was, can an inosine-containing aptamer still be converted to a biosensor? We do know that the MNS 4.1 aptamer, the parent aptamer, has been converted into multiple different types of biosensors. But what happens to that biosensing ability if we change G to I? And so we generated structure switching aptamers. So just briefly, I know that structure switching biosensors out of these aptamers. I'm sorry that not only am I introducing aptamers to people who aren't used to them, I'm so sorry that I'm introducing structure switching biosensors as well. But basically, again, as I stated, we Sci5 labeled our aptamer for MST. Taking advantage of this Sci5 labeled aptamer, we also generated a partial complement with a quencher on it. I will refer to this as our capture strand. In the absence of cocaine, of course, those two strands are going to hybridize together, bringing the fluorophore quencher into close proximity, leading to fretting, and then we lose our fluorescence. We quench our fluorescence. In the presence of cocaine, however, that quencher strand is displaced, and we then regain our fluorescence in the presence of cocaine. This can be done for essentially any aptamer that has 2 or 3 stems, and can have some type of structure switching ability. So this is something that really can be done for any aptamer that's out there. From here, we are able to generate the percent displacement. So we get a percent fluorescence, we back calculate to get a percent quench, and then from there, we back calculate again to get a percent displacement. That tells us how much cocaine is bound. And then from that, we were able to generate successfully not only a structure switching biosensor for cocaine, or with MNS 4.1 aptamer, or different type of structure switching, we were also able to still generate it for the MNS 4.1 inosine aptamer too. So we were really, really excited about these data as well. And with that, I would like to thank my group, especially Dr. Amy Sanford, who just recently defended, but she is a co-author on this paper, and also my best friend and my lab mate, Sierra Sterling, who is my other co-author on this paper. Of course, our funding and my lab and my PI. And with that, I'll take any questions.
Fantastic. All right. So let's go ahead and just jump straight into the questions. And anybody that, this is not a cutoff for questions, so certainly feel free to add more as we go. So let's see. First question is, how do you ensure that you're selecting for your desired target once you've narrowed down the candidate? So you kind of referred to, I think the terminology was specificity. Is that right?
Celex, yeah.
Okay. And so basically, like, how do you know that you're selecting for the actual target that you want, versus something that's, say, similar but
yes,
not what you're interested in?
Yeah, great question. So we do selection, but we also do negative selection. So you do the same thing with Celex, and let's say we want cocaine binding aptamers, we would introduce norcocaine because it's a similar analog. And those sequences that are active, we get rid of and we carry forward with the rest of them.
Okay.
So we perform a negative selection with any off target to get rid of those. And then we know that we have aptamers for those because, again, you can do some type of binding study with your aptamers that you generate and then your target, and you can see if you get a KD.
Okay, that makes sense.
Yes.
Let's see. So with MST, are there any concerns with damaging the aptamers or causing unintended changes when you actually heat
them? That's a fantastic question. I don't know that heating them up is the actual issue there.
Okay.
I don't know that it gets hot enough to heat them up. That is a great question. I think the actual issue may come into covalently labeling them with a 4-4. So the thing is, we don't see a huge perturbation in it, but it does not mean that a less stable aptamer that actually is less thermostable as well may not run into that issue. It is definitely possible.
Okay. All right. I mean, that makes sense.
Yeah.
Let's see. Is the group interested in exploring other nucleic acid-based aptamers that aren't DNA, or are DNA-based aptamers the better choice?
Depends on your application. I don't want to say yes, DNA is better. It definitely depends on your application. DNA is a lot more stable. DNA is also a lot cheaper to generate than RNA. So our group does work with both RNA and DNA aptamers. So yes, both are okay. I just think that DNA is a lot more stable, a lot easier to work with. You don't have to worry about breathing on your DNA sample. You breathe on your RNA sample, you can get rid of it. Yeah.
Okay. Yeah, no, that would be an issue for me. That's for sure. That makes sense. So I guess that means that DNA is also a better option for, like, I don't know if you guys are really concerned so much with this, but like if somebody was looking to commercialize this, I guess it sounds like DNA would be the better choice for the reasons that you just gave.
Yeah, there are ways to get around RNA degradation as well. You can have modified bases. So basically, instead of a sugar backbone, you may use a modified backbone that that RNase may not recognize, but you still run into the money issue because not only is RNA more expensive, RNA with modified bases are even more expensive.
Makes
so
sense.
Ideally, DNA, yes, is typically the way to go. That's actually what's used a lot too. You can use it over and over depending on your application, versus RNA at some point will degrade much quicker than DNA.
Okay, that makes sense. Let's see. Can you monitor the rate of structure switching, and does the structure switch when exposed to different substrates?
Monitor the rate of structure switching, and what's the other question? I'm sorry.
Does the structure switch when exposed to different substrates?
Great question. We did not test other substrates, but because it binds other substrates, I'm pretty sure it would switch as well. It may, because it has a lower affinity for some of the substrates, it may not be as efficient, but I do think, yes, we would still kick off that quencher strand because the aptamer would still want to bind that other substrate. As far as measuring the rate of structure switching, it's not something I've done, but I don't see why you can't. It's not something I've done in lab.
Okay. Let's see. So just somebody wanted to, Dr. Spate wanted to say that it was a great talk, and I agree. And I guess I'll ask one more question, just sort of generally speaking. So you're showing that you can generate a range of responses to, in this case, cocaine, or some of, I guess, I don't know if derivative is the right word. This is where my chemical engineering comes in hard. But you showed that you could do a range of like binding affinities. What does that let you do? Like, I don't want to say what's the application of it, but what does that allow you to do in a more practical sense? Like what's the bigger picture interest in that?
Yeah. So as I mentioned, ironically, this was actually a part of another project that my PI did give me permission to talk about, but I won't talk about that because I feel like it gets like super deep into the biochem.
Okay.
But if you, again, let me go actually to one of the pictures. If you have someone who, again, cocaine is going to hydrolyze in solution. Cocaine is also going to hydrolyze in your urine, or in your body, because you have enzymes that are going to break it down. You have esterases that want to hydrolyze that bind, right?
Yeah.
So in the case that, let's say you have a drug test sample from one person, a drug test sample from another person. One person had done cocaine, let's say 24 hours ago versus 12 hours ago. You would probably want a more sensitive aptamer for that 24-hour sample versus the 12-hour sample. Okay. So it gives you the opportunity to test older samples, for example, if someone would ever use it as a biosensor. We did not go as far as to test it in actual human samples. Okay. But if you were to want to test someone who had, again, done cocaine much longer ago, is that the right? Yeah. Or much earlier in the day than one other sample, you have the opportunity to use a more sensitive aptamer versus not being able to ever detect it because too much time had passed, for example. Okay, I got you. So it gives you a little bit finer detection, basically. Yeah, exactly. It gives you a better limit of detection. And then also, it's just much quicker if you can do this by changing G to I versus going through CLEX again or truncating and crossing your fingers. I got you. You can simply order these. Yeah, you can simply order these with a bunch of different G to I substitutions. And get all of your aptamers in an order and get a range versus just truncating and really, really hoping for the best. I got you. Okay. Well, that actually kind of your answer there kind of leads into this questions. On the slide you show the ability to swap the G to the I. How easy is it to be selected for which G, or is it semi-random? And can you select an exact G? That's a great question. You can select an exact G because you can just simply order it. We order from and sometimes from the University of Utah, but when we send them the sequence, we just type it from 5' to 3' into the database, and they synthesize it and give it to us, and we can, again, just choose any G you want. We went for STEM 1 because we knew that STEM 1 was very, very important for binding, and because that's what other people, went after in literature. And we also wanted to choose far away from the binding pocket just in case it would perturb the binding drastically. But I would say, you would just look at the structure of your aptamer specifically and maybe study the binding patterns if it's not already in literature, and then choose your G's based on that. Or you can just, choose a random G and see what happens. Okay. So are there other instances where you would actually want to modify the binding pocket to, say, increase the limited detection, or maybe for some reason decrease it? Are there any examples that you know of where that's something that's done? Yeah. So we don't specifically target the binding pocket for this reason. What I'm doing for this project, or what this project was used for, is we're looking at how aptamers can also help increase enzyme activity, which I cannot wait to talk about in a later talk. Looking forward to it. Yeah, but as you know, enzymes have a KM, which isn't like it's not the KD, but it's similar. It's still how well the enzyme recognizes its target. Okay. In our case, we want an aptamer that has a similar KD to the enzyme's KM. And when we realized that at the time, in the conditions that we had, when we realized the aptamers were too good, so when we realized all of these were better than our enzyme KM, we considered looking at the binding pocket to go ahead and get a worse aptamer. We looked at G's like here as a way to get an aptamer that had a worse KD so that we could be closer to our enzyme KM. Okay. So it would really just depend on your application. Okay. And so I'll ask one more question, and like I said, if anybody else has any they want to get in, please feel free to do so. Again, this is my ignorance of the field. Do you have to, when you're making these changes, so like swapping the G's to the I's or any other changes that you make, do you need to have any concern for like biocompatibility or anything like that, or is that not really a big concern because I guess it's mostly done outside of the body? I guess, right. I'm trying to ask this in a not dumb way. Yeah, like are there any concerns that you have to keep in mind, aside from obviously like the binding affinities and things like that when you're making these swap outs? As far as putting it in the body, no. Inosine is in nature. Typically in RNA, A is converted to I by an enzyme. So it is in nature. It's just in that case is bad, but in this case, no, it's biocompatible because it's not far off from guanosine. As a matter of fact, the body will actually recognize inosine as guanosine. The body cannot tell the difference. Yeah.
Interesting. Okay, I did not realize that.
That's cool. So I typically wants to naturally base pair with C, just like G would.
Okay, I got you.
Yes.
Okay. No, this is really cool stuff. Like this is super cool work, and I can't wait for you to come back and present on the full story that you've got that you're finishing up now. So it looks like that's all the questions we've got. So I'll thank you again, Brea. This was, like I said, super cool, really interesting talk. You're doing amazing work. It's really cool to learn about. Thank you to everybody for coming out. I really appreciate it. Thank you for the great questions. And we will be back next week. So I hope you'll come back and join us then. And so in the meantime, have a great weekend. Stay safe. We will see you next week. And Brea, if I can get you to hang around for just 1 second, we'll talk briefly, and then I'll wish you a good night as well. So thanks again, everybody, and see you next week. Have a good night.
Thank you so much.